Traditional hand transfer methods of bulk reagents into microtiter plates are slow and prone to human error. This technology more than doubles the efficiency of the process and helps eliminate that error.
This technique can help users understand operations performed in microplates, reduce opportunity for errors, and increase user efficiency during manual pipetting operations.
This technique will positively impact outcomes in preclinical and clinical settings by removing and reducing mistakes that can be costly and even life-threatening if not corrected.
This open source system can be easily adapted for other applications, such as microplates copy inspections, to eliminate inconsistencies in either compound or bioassay placement for chemistry and biological needs.
Demonstrating the sample transfer procedure will be Lena DeLuca, a compound manager from the Lead Identification Laboratory.
To set up a semi-automated plate-to-plate sample transfer, use a spreadsheet editing application to generate a CSV file containing source and destination plates, labeling the columns source barcode, destination barcode, source well, destination well, and transfer volume. Under the columns, include one row in the file for each desired pipetting operation, indicating the alphanumeric barcode of the source microplate if there is one, the alphanumeric barcode of the destination microplate if there is one, the alphanumeric row and column identifier for the well to be pipetted from in the source plate, the alphanumeric row and column identifier for the well to be pipetted from the destination plate, and the volume to be transferred from the source well in the source barcode column to the destination well in the destination barcode. Open the Light Guide program to open the Microplate Assistive Pipetting Light Emitter Plate-to-Plate Graphic User Interface application, and click Select Cherry Pick File.